to Progressive Education Society's Modern College of Pharmacy, Nigdi, Pune. In this video, we will discuss about the morphology, microscopy, and microchemistry of Rauwolfia roots. Myself, Dr. Bhushan Pimple. Rauwolfia roots are commonly known as sarpagandha in India. These roots, these are the medicinal agents are not only derived from roots but also rhizomes. So the drug, a commercial drug, it consists of dried roots as well as rhizomes of Rauwolfia serpentina benth belonging to family Apocynaceae. The major chemical constituents are alkaloids, and the major alkaloid is rezapine and ajmalin. Besides, it also has certain other alkaloids which have been found to have beneficial medicinal effects like ajmalicin, serpentine, serpentinine, and resinamine and yohimbine. The basic reason why Rauwolfia is used medicinally is its antihypertensive nature. So it decreases the elevated blood pressure. And in traditional systems of medicine, this sarpagandha is used for treatment of various neuropsychiatric disorders or mania. Ajmalin present in the roots has been found to be effective in the treatment of arrhythmia, very similar to quinidine. Yohimbine nowadays have been extensively exploited or used for the treatment of erectile dysfunction. Now, erectile dysfunction is the major reason why yohimbine is sold and in the commercial markets, and it is very similar to Viagra. The color of these roots are externally it appears grayish yellow or light brown, and when the bark is taken off, the inner wood appears to be light yellow in color or creamish in color. Generally, the fresh roots they have some characteristic odor very similar to that of any crude drug or one aushadhi. But if you store it for a period of say suppose 2 or 3 months, the odor vanishes. So it becomes odorless. Taste is bitter because of the presence of more than 40 alkaloids in it. Usually in the commercial markets, the root cuts are available in the form of around 2 to 10 centimeter in length and 0.5 to 2 centimeter in width. Shape is cylindrical and slightly tapering towards one end. Now the major diagnostic feature of this root is its tortuousness. Tortuousness means the root is appearing very similar to that of a snake, a wavy appearance. And special feature lies in its external appearance. When the roots are too young, they give out slight wrinkles over the surface, whereas upon maturation, the roots show prominent longitudinal ridges or furrows. Now, these are the roots, and even if you take a smallest piece, it will have some tortuousness or wavy appearance. And if you want to take out the section, the root is held in this way, and the roots have to be soaked overnight before taking section. And sometimes it may not become softer even after soaking overnight. So you can boil the root with water and the boiled root, here, once it becomes soft, are ready for cutting the sections. The sections are cut and the cylindrical root gives out ring-like pieces. The centermost region is xylem and the external cream color is phloem and cortex, whereas the extremely outermost region, which is brownish in nature, is the bark of the root. So even if you want to see the location of these tissues, there is no need to have a microscope for the detailed study. Yes, you require a microscope, but a thick section where on staining with fluoroglucinol HCl will give this kind of appearance. So you will be able to understand where exactly the wood portion starts. Now, these are the sections of root, and in root you will not find the pith region. Pith is present in rhizomes. So how to differentiate between roots and rhizomes is by taking a thick section and dipping it in fluoroglucinol HCl. If you find a whitish color spot at the center, that indicates the presence of pith. That means it is rhizome. And if there is no white color spot at the center, and if the center is completely filled with pink color, this indicates it is a root. So we will just have a look about of the layers of the tissue present in the Rauwolfia section. Once you take the section, I have highlighted only a small region of the circular section. The external tissue is stratified cork. Now, why are we calling it as stratified is because you will notice the presence of certain tissue or certain cork cells have been stained reddish in color, whereas others have not yet stained. So the layers are alternate in arrangement, and it gives a stratified appearance, very similar to a stratified epithelium. This appearance will be visible only if you stain the section with fluoroglucinol HCl. This indicates some of the reddish colored layer of the cork cells are containing lignin, whereas others do not have. And the arrangement of lignified and non-lignified cork cells is alternate in nature. The next layer is phalogen. Again, it is quite similar to cinchona. Phalogen and phalloderm are indistinguishable. You cannot distinguish between the phalogen, phalloderm. But yes, cork can, after staining with fluoroglucinol, you can easily mark the cork. And if you stain it this section with Sudan red 3, the entire phalogen, phalloderm, and stratified cork or the cork, it appears pink in color because all these three layers are containing suberin in them. The next layer is cortex region. Now, cortex is the layer in root or stem which is specially designed to store the photosynthesized food product. And plants, they store their food in the form of starch. So if you cut or if you stain this cortex with iodine, you will notice certain purple or violet color, light purple or light violet, or sometimes bluish color starch grain located in the cortex zone, as well as certain calcium oxalate crystals can also be visible in the cortex. The next is secondary phloem region. Now I have highlighted or I have shown here a very short layer of secondary phloem. As such, it is larger layer of secondary phloem. Again, secondary phloem is functioning in storage of food material, and it gives out radial cells which are called as medullary rays. You can see here, these are the medullary rays which have radial elongation of the cells, and they traverse from center towards the secondary phloem. Now the role of secondary this medullary rays is to transport the food material from the center towards the cortex for storage of the transported food material. And the innermost zone in case of root is xylem. This is completely lignified and it has large vessels. These large cells or cells like appearance are vessels of xylem, and one thing can be noted here that the xylem vessels are border pitted. Border pitted means it is a type of thickening provided to the xylem vessel. Thickening in order to maintain its rigidity. These are just large pipes which are flexible, and the externally it has been coated with some lignin material, and very small dots or pores are left out, and this gives a border pitted appearance to the xylem vessel. So the major diagnostic feature of Rauwolfia is its stratified cork and border pitted xylem vessels. Rest all the roots will show very similar anatomical structure. As far as staining is concerned, if you use Sudan Red 3, as I have said, the phylloderm and cork will get stained pink in color. And if you use fluoroglycinol HCl, the color of the TS will appear as you are watching right now. The uppermost layer will shows the cork will show stratification, whereas xylem will show complete pink coloration. Besides, you can also use acetic acid for clearing out the unwanted material in the cell or cellular debris, and in order to highlight the calcium oxalate crystals. Mostly the calcium oxalate crystals are prismatic in nature. They provide rigidity, or they are derived out of excretory product called as oxalic acid. Now, this is the real image or photomicrograph of Rauwolfia cork. Now, you can notice here the cork has layers of lignified as well as non-lignified layer. Strips can be observed here on staining with fluoroglycinol HCl. This image is taken at 10x objective and 10x eyepiece. Now, if you zoom in, or if you take a look at 5x objective, you will notice the location. The yellow color indicates cortex and secondary phloem, whereas the pink color region at the center indicates the xylem. Even you can notice the cork at this outermost periphery here. So this is how it appears. Now we will look at the external layers of this anatomy of Rauwolfia root, and it comprises of periderm. Periderm has three layers: cork, phyllogen, and phylloderm. Now you can notice here the cork has been stained with fluoroglycinol HCl, and the strips can be observed clearly here. The white color cells which are allowing the light to pass through them are non-lignified cells, whereas the certain cells are arranged compactly and they do have more amount of lignin in them. So they are showing a strip or a band-like appearance. That is the reason why this cork is called as stratified cork. Just below that, in none of the tiers, you will notice the clear presence of phyllogen and phylloderm, and that is why it is indistinct. You cannot distinguish between phyllogen and phylloderm. And just have a look at the layers, how the cork cells are arranged. They are stacked one above the other and not in a staggered fashion. They are stacked one above the other like a stack of coins or boxes. Yes, here you can notice the arrangement of the cork cells one above the other. So this is how periderm appears upon staining with chloroglucinol HCl. The region is cortex, and as I have told you, cortex is lying just below the periderm. So the lower region wherein you can find golden yellow color containing round globules is the starch grain and the cortex parenchyma. Let us have a look at another image of this cortex. Here, the round starch grains are clearly visible, and each cell is stacked with more amount of or abundant starch grains are present per cell. Yes, this is the fine TS taken, and you can notice the presence of the starch grains. These are not calcium oxalate crystals, but these are round starch grains. If you stain these starch grains with iodine, you will notice light blue or purple color getting absorbed into the iodine getting absorbed into the starch grain. The next layer is secondary phloem, and you can differentiate between secondary phloem and cortex by the presence of large diametric cells. Have a look at the location of secondary phloem. Secondary phloem is present above the xylem, and it gives out branches which traverse through the xylem and reach the center. Now this is the section of a rhizome, and why am I calling it as rhizome is because look at the lower left bottom. It has pith region, which is very similar to the secondary phloem, the color. This means it is a rhizome. And you can find the connection between the pith and the secondary phloem by medullary rays. This is the longitudinal section of the root, which indicates the presence of this xylems with xylem vessels having border pitted in sense dots can be observed on the xylem vessels. Yes, this is the transverse section of the root taken under 40x objective and 10x eyepiece. So the large hollow regions are called as xylem vessels, and the gray or light black color radially elongated cells are medullary cells or medullary rays. These are medullary rays. This is xylem vessel, and the entire rest of the part is covered with xylem parenchyma, which gets stained immediately on staining with chloroglucinol HCl. This is another photograph of the same region, xylem vessels and the medullary rays and xylem parenchyma. The secondary phloem can show or cortex can show the presence of calcium oxalate crystals. Calcium oxalate crystals are sometimes prismatic in nature, or the shape varies from parallelogram or rectangle or rhombus. This is the way how the medullary rays gets converged towards the center. They are very similar to a radius of the circle. From the periphery, they are getting converged towards the center. And I've stained it with iodine to make the starch grains visible clearly. So you can notice the starch grains are traveling from the center towards the periphery through these medullary rays. Again, the white colored medullary rays. Yes, this is the TS of rhizome. Why rhizome is because once the medullaries and secondary flow xylem has uh, completed or has finished, just at the center you will notice large such cells. Large cells indicates the xylem region. Oh, sorry, pith region. Pith is the soft part which is present located at the centermost zone of the rhizome. So this allows the food to travel and then pushes the food through these medullaries into the secondary phloem or cortex region. So this is all about Rauwolfia root and rhizomes.